Hello friends, welcome in my classroom, and in this class we will learn functions of plasma membrane. So the plasma membrane, it allows the required material to put in the cell and unwanted or waste material to take out from the cell. And this process are done by the two methods, that are the diffusion and the osmosis. So firstly we will learn about the diffusion. We will understand the diffusion. So to understand the diffusion very well, we will take an example. Have you observed any time as the time of the summer when we take a glass of water and put the some amount of roux afza in that glass of water? Don't disturb the glass of water, and after some time you will observe that the roux afza automatically mixed with the water. So what is that? That is a kind of diffusion in which the higher concentrated roux afza get mixed with the lower concentrated water molecule. So not only the substance like the roux afza get mixed, the oxygen, carbon dioxide, water, they are also diffused in the cell. So we have taken the example of the roux afza and the water. Similarly, in the cell, this carbon dioxide, oxygen and the water, it also diffused from the higher concentration to the lower concentration. Now we will understand here. Here you are seeing the higher concentration of the molecules, and here it is the lower concentration of the molecule. So the movement, it is from the higher concentration to the lower concentration. We can understand more very well with this diagram. Here it is the amoeba, and in this amoeba requires oxygen to respire. So the oxygen present in the water body or in the water, it diffused through the water. The oxygen diffused through this cell membrane, and through the process of the respiration, amoeba releases carbon dioxide, and that carbon dioxide, it expelled out into the water bodies, and this process is called the diffusion. So friends, now you can understand very well how the diffusion takes place in unicellular organisms. Now we will learn about the osmosis. So the osmosis, it is also a type of the diffusion, because in the process of the osmosis, the water moves from the higher concentration to the lower concentration, and in this diagram you can see this layer that I am showing you. This is the semi-permeable membrane. And semi-permeable membrane, it is also called the selectively permeable membrane, that allow only the selective material to pass in or pass out. So in that diagram, here the one side, here it is the water, and these are the larger particle you are seeing. They are the solution of the sugar molecules. So here, as the one side that having the sugar solution, so the water concentration on the left, So the water concentration on the left, it is higher than the water concentration on the right. So the water, it moves towards the higher water concentration to the lower water concentration, and this is called the osmosis. So the osmosis, it is also depend on the types of solution. And how the types of solution define the osmosis? Let's see the type of solution that define the osmosis. So let's understand about the types of solution. The hypotonic solution. The hypotonic solution, it is a type of solution in which the cell put in higher water concentration, means the water concentration higher than the cell. So whenever the higher water concentration is outside the cell, then the cell start gaining the water and swelled up. So the such kind of solution is called the hypotonic solution, in which the cell gains the water. And when the swelled up cell, when put in the solution that having the lower concentration, lower water concentration than the cell, then cell get shrink, means the water present inside the cell, it releases from the cell, and the type of solution, it is called hypertonic solution. And in plant cell, when the water releases from the plant cell and move outward and cell gets shrink, then the cell is called the plasmolysed cell. Then cell is called plasmolysed cell in plant cell. But when an animal cell like the RBC, it gets shrink, or the water concentration that release from the cell, then that cells are called the crenated. The animal cell, they are called crenated. So when the animal cell gets shrink due to the hypertonic solution, then they turned as the crenated, and when the plant cell gets shrink due to the hypertonic solution, then they turn as the plasmolysed cell, or the process it is called the plasmolysis. And now the isotonic solution. It is a type of solution in which a cell capped and the concentration inside the cell and the concentration of the solution, it is same. So there is no effect on the cell, and the type of solution is called isotonic solution, where no change on the structure of the cell.